All right, so this last video is just going to be talking about a little summary of when cells would need to do fermentation and talk about really what fermentation is. So if a cell has a mitochondria and there isn't oxygen available, most of the time it's going to want to do cell respiration. But there are times when it's not available, as well as some species can only do glycolysis. And it's really what they do with that. What is the byproduct of pyruvate and how does it get converted back is going to determine if it's called anaerobic respiration versus fermentation. So let's talk about the difference between them real quick. If we say it's anaerobic respiration, typically going to be found in anaerobic prokaryotes. So that we're not necessarily going to say it's fermenting. It's going to use glycolysis. Oxygen is not the final electron acceptor. Again, the big thing here is that regardless if it's fermentation or anaerobic respiration, you're not using the mitochondria and it's way less efficient. You're only, okay, I'll even say that both of these, it's only substrate level phosphorylation. Basically means, yeah, you're making ATP, but it's not the most efficient way. But it's better than nothing. So let's talk about this. Like I mentioned in the previous video, if you didn't watch it, there's little channel proteins here. And basically that means is if oxygen is there, these channel proteins are open. That lets pyruvate know, okay, it's kind of an all or nothing. Oxygen's there. We're actually going to go ahead and do the Krebs cycle, the electron transport chain. If it's absent, this thing closes. And pyruvate is going to go and either make ethanol or lactic acid. It depends on the enzymes, which again depends on the species. So the DNA is going to be determining on what enzymes are produced. So pyruvate is going to go and either make ethanol or lactic acid. But regardless, is this first step is glycolysis. So it's the first step in making ATP. That happens whether you're doing lactic acid fermentation or alcoholic fermentation. So glycolysis is kind of the ATP production. But let's kind of get into fermentation, whether you're doing alcoholic or lactic acid. So let me kind of start this off by just saying, what is the point? The point is you need a way to regenerate NAD+, right? Because if you forgot from the last video, all that NADH is going to be used to actually power the electron transport chain. Well, if that's taken away, there needs to be a way to regenerate NAD+. NADH needs to dump their electrons off somewhere, okay? So that's kind of the point, is NAD+ regeneration, okay? Because you need NAD+ for glycolysis, and that's what this is showing here. Glucose makes pyruvic acid. You do make some NADH. And if you're not necessarily doing the whole respiration in terms of aerobic respiration, this whole process is done, right? So you need a way to actually remake the NAD+ to pick up the electrons, okay? Now again, all cells can do this. No oxygen is needed, so all cells can do glycolysis. Now, this just kind of shows you the difference between alcoholic versus lactic acid. Not something that you got to get crazy specific with, but you do at least need to understand this first part, right? So glucose into pyruvate, this is just good old glycolysis, right? Nothing new here. But once you make pyruvate, it can't go to the ETC, so basically it loses some CO2. You make what's called acetaldehyde, you make ethanol, and that's interesting. You do make alcohol as your byproduct, but the key is all in right here. You're remaking this NAD+, okay? Again, why does that matter? Well, if you can't make NAD+ again, you're going to eventually stop glycolysis. So common test questions is, well, what if you can't regenerate NAD+? Well, then you can't even do glycolysis. So normally, like we mentioned earlier in the previous video, NADH wants to dump those electrons off to the ETC, the electron transport chain. But if it can't, it needs a way to at least remake NAD+. So it's a quick way of dumping off electrons. The electrons from NADH get dumped off, add acetaldehyde, and then that makes ethanol as your byproduct. So that's the point. The point is you're remaking NAD+ to allow it to redo glycolysis. So what is the difference between that and lactic acid? Well, not a whole lot, in the sense that what you guys have to know, again, glucose to pyruvate, that's just glycolysis. the electrons get dumped off on pyruvate, and then that makes lactate or lactic acid as a byproduct. So if you think of it in terms of like human muscle cells, your body, if you're working out a lot, your body actually switches over into lactic acid fermentation because your cells are using more ATP than needed. There's no oxygen. So it quickly switches over, makes lactic acid as a byproduct because it needs ATP quick, and there's no oxygen available if you're doing really anaerobic exercises. So in terms of that, this is kind of a nice little recap of the three: aerobic, respiration. Okay, that's just good old cell respiration. You do have a mitochondria. The final electron acceptor is oxygen, and about 36 to 38 ATP, depending on if oxygen is available. And again, this is starting out with glucose. If you start out with glucose in anaerobic respiration, no O2, it occurs in the cytosol. It's an alternative for your electron acceptor. ATP production is going to be produced pretty slow and not nearly efficient. Fermentation, no O2, happens in the cytosol. Again, no mitochondria is needed for either one of these. It's just really what's the byproduct. So what is the evolutionary significance about this? It's basically and this is kind of key, it's the first way cells were generating their ATP sources, because remember the early Earth didn't have an O2 rich atmosphere. The Miller-Urey experiment will explain that. Basically, that means that without O2, you can still make ATP. So it's likely the first way that all of us were able to actually make some ATP. Again, you don't need a mitochondria, so that means way back in that endomembrane, endosymbiotic theory, they all essentially could make some ATP without a mitochondria. Okay, and again, that is a massive key that for the endosymbiotic theory. So in terms of this, this is just kind of recapping what I was mentioning earlier in one of the videos. You can eat proteins, and proteins can jump in and at different steps to still make some ATP via oxidative phosphorylation. So it's not just glucose that is your starting reactant. Now, the last thing that I do want to mention here is how is ATP regulated? And again, regulation just literally means control. How do we control ATP synthesis? Well, ATP can actually bind allosterically. If you've got allosterically means it can actually block off the functionability for one of those first enzymes in glycolysis, phosphofructokinase. So basically that means if you make too much ATP, if the cell has enough, that ATP can bind to the enzyme and shuts it off. Okay. Now on the contrary, AMP actually acts as an allosteric activator, which basically means adenosine monophosphate. After all, your ATP has been used up and it makes adenosine monophosphate. Adenosine monophosphate, AMP, can actually turn on this enzyme and it starts this whole process. So you have an allosteric inhibitor, which is ATP. And again, you think about it, it makes sense. If it doesn't mean like one glucose makes one ATP, it shuts the whole thing down. You have to have a really high concentration for this to take place. But it's really interesting when you think about what's happening here. Too much ATP, you don't want to overproduce it, so it inhibits it. But once all that ATP is used up, because remember, ATP, when it's used, gets converted into adenosine diphosphate and eventually adenosine monophosphate. So it lets the cell know that, hey, AMP is going to turn on this whole pathway. So that is kind of the last little bit of chapter 9, which was on respiration. I want to show you this quick little overcap of really what fermentation is. And this just kind of shows you another pathway where NAD plus just needs to get regenerated in glycolysis. So again, that is the bulk of chapter 9, and I hope it's helpful.
In a process called fermentation. By oxidizing the NADH in glycolysis, fermentation regenerates NAD+, which can take part